It's a really great pleasure to be here, and it's quite exciting for me to be able to talk in front of this small, intimate group. It's really the presentation style that I prefer because it gives you an opportunity to ask questions that you may have. And really, you should feel free, if something I'm saying is unclear, please interrupt me at any time so that I can clarify what I'm saying. For more in-depth questions, you might want to save them to the end of the talk, because I've already anticipated a number of your questions that you may have, and I'll try answering them at the end of the talk. And if they're not answered there, then please ask, because I don't want anyone to leave without feeling that I've answered all of your questions. And also, I can stay around as long as anyone needs to answer your questions. So interrupt me during my talk if there's a point you're not understanding. And then, like I said, there'll be plenty of opportunities to ask me questions towards the end of my talk or even after the talk if you feel more comfortable doing that. I have to start by saying it's just a wonderful to be in this country. Ireland is fantastic. I don't know why people complain about the weather here. It's great. It's really, really nice. I also need to send out special thanks to several families who have embraced my family in my visit here. So I've actually taken quite a long trip here, a total of 3 weeks. And for the first 9 days, we had free accommodations thanks to parents of individuals with Angelman syndrome. And I think that's a wonderful gift that you could give to me, and it's greatly appreciated. I wanted to do some specific thanks, if this is going to decide to work or not. All right, I'll advance it this way. First, I want to thank Sarah, because we had a wonderful time in Leitrim and staying at your place there. We use this as a jumping off point to make a number of trips. This is my girls in Carrowkeel, which is to go to something that's 3,000 BC and be able to have kind of unabated access to it. It's amazing. And the hike up there was just striking. And then we love going to Loch Key. Of course, I'm drawn to the castles. I couldn't get my girls away from the playground, of course. Then we went up to Downings, thanks to Aisling. And once again, it was incredible, just right out their back door at their place there is the ocean. Because they was able to loan our children wetsuits, they actually got in this freezing cold water and enjoyed themselves immensely on their bodyboards. I particularly like the hike up to Horn's Head and just the cliffs there. I've just never seen anything like it. I mean, they just plunge for hundreds and hundreds of feet into the ocean. It's quite striking. And I let my girls slowly belly out to the end and peer over, and I was always holding on to them, except I told them to stay there for 5 seconds so I could step back, take a picture, and run back and grab them. But it was fantastic. And then thanks to Sarah for letting, well, her dad actually let us stay at his place, which was great. You can see my girls playing on Clara's bikes there and having a blast, and I had a hard time getting them away from the house, because the house had such a great backyard with a wonderful garden and toys and playhouse that they didn't want to go and leave the sites, but we were able to drag them out some. And one of my favorite places to visit was Kong, which is just a wonderful city. This is the old fisherman's hut along the beautiful river there. But now to jump into the science and to change gears a little bit, I have to state very simply that the goal of all of this research in Angelman syndrome is to make a path forward to improve the lives of individuals with Angelman syndrome. So without a doubt, no matter what we're doing, we have this end goal in mind. Now, there are a lot of different paths to achieve this and towards developing therapeutics. And one of my goals today is to help you appreciate those different paths and why we might take different paths. Now, I think the one that most parents are interested in, and actually what most parent foundations fund, is that of drug discovery. Okay? Now, there are a number of reasons why, as family members, you're particularly excited about this and why there's a lot of attention for this, and that's that it has a clear advantage. So it has the potential to be relatively rapidly translated into a usable therapeutic. Okay? So that's the great goal of this and the advantage of this type of therapeutic approach in developing drugs. It also has some clear disadvantages that I want you to know as well and appreciate as well. One, that it's risky. So it's risky in two ways. So most drugs and compounds that are brought forward fail. So even in the best thought out ideas, even with the might of a pharmaceutical company behind them, when a new compound is tested, less than 1 in 10,000 succeed in making it to market. Okay? It's also risky because eventually you have to try a compound, a potential drug in an individual. And of course, there are risks that it might not behave as it had in an animal model, for example. So there's a risk associated with that as well. It's also costly, and this is something I'm going to get back to. So I think there's an expectation that drugs can be very cheaply developed. The fact is the average cost for a new FDA-approved compound to use by individuals is $1.5 billion. Okay? Now, has another advantage, and that disadvantage is these are often one-off type trials, that if a compound is taken forward and it doesn't work, then you haven't really gained much information that's useful to bring forward other compounds. So it's often the case that drug companies don't look into why the drug failed, how the drug was working or not working like you might expect. And if you don't explore these aspects, then you've really gained no new information. So you're back immediately to where you started at ground zero. Okay? Now, another path forward is basic research. And my lab takes a two-pronged approach. So roughly half of my lab is working on drug discovery. The other half of my lab is working on basic research. And I don't think often that many parents appreciate the importance of basic research. So I wanted to take a little time and tell you why this is also very, very important to what we do. So one of the advantages, this is a very steady approach that provides foundational knowledge that can be useful to every other approach to develop therapeutics for Angelman syndrome. So all the knowledge we get from basic research provides a foundation upon which we can build, and it'll help every other therapeutic approach. So the insights that are gained here can guide other approaches for developing therapeutics. Now, the drawbacks are also, of course, obvious. Well, it's slower. Okay, so it may be a sure way forward, but it's much slower. And it obviously does not directly test therapeutics. Okay? Now, I wanted to, before I talk about what we're doing for drug discovery, which I think is traditionally what has interested the parents the most, I wanted to tell you just briefly about why we do some of the basic research experiments that we do, because I think they're often misunderstood by some parents or why they're important. So one such experiment that we're doing now is to determine the expression pattern of UB3A in the brain across development. Of course, you all know that UB3A is the gene whose function is disrupted that causes Angelman syndrome. So why would we want to, in a very detailed way, understand where UB3A is expressed in every area of the brain and when it's expressed and what its expression patterns are? Well, to you, it might not make much sense that we're putting effort into doing this, perhaps, because it doesn't immediately develop a therapeutic. But I would try, maybe by analogy, help you to understand why we're doing it. And one is, you can imagine me coming to Dublin for the first time, and someone tells me, well, you should really go check out the Guinness Brewery. That's great. What if I don't have a map? How long is it going to take me wandering around this area before I come across the Guinness Brewery? Well, I'm going to be wandering for a really, really long time before I find it. If someone gives me a very, very rough map, it might really still take me a relatively long time, although it certainly help. It'll still take me a long time to find the Guinness Brewery. Now, if someone gives me a very, very detailed map, well, then I have a path forward. I know where to go, and it's just a matter of going down that path. And that is a little bit why I think that looking where in the brain UB3A is expressed and when it's expressed and how it's expressed it's important to neuroscientists. So as a parent, this might say, okay, this is not the development of a therapeutic. Why would you do this? And as a scientist, I say the answer is pretty simple. It's giving us a roadmap forward for the development of therapeutics. So here, I like for parents to appreciate some of the raw data. So I try to weave it into the talk. This is an example, and it might be hard to see. I don't know if we can dim the lights a little bit. Here, let's see if I can do it here. There we go. These are 3 different sections of a mouse brain, and we're looking at UB3A staining. And the fluorescence, the green fluorescence, is where UB3A is. Now, these are 3 different ages. This is postnatal day 0, so right at the time of birth. This is at postnatal day 7, which might be a kind of a toddler age child equivalent, and postnatal day 28, which would be the equivalent of a young adolescent. Now, first of all, you can see the brain is growing, so obviously it gets thicker and thicker. But to you in the audience, does the expression of UB3A look the same across development? No? What looks different about it?
It's fainter.
It's more spread out, it's more faded. So it's really different. So if you look at a higher, let's see if I can get this to work. If you look at if you look at a higher magnification, you can see that the expression pattern really changes. So early on in development, it's in the cell body of a neuron and all of its processes. Okay? Later on, it's more restricted to the nucleus, so the very center of a neuron in the brain. Now, once again, it might be harder for a parent or a caregiver to understand why this is important. If you can advance, from a parent perspective, this tells me something important. It says that the forms of UB3A that are being expressed might be changing across development, and this is something that I should really look into. So for example, if you're doing a gene therapy where you're trying to reintroduce a specific form of UB3A, you might want to make sure you get the right one. Now, if you could advance, it also might say, well, the functions of UB3A might be changing in age, because early on, it's expressed all throughout the processes of the neuron, and then later on, it's expressed more in the nucleus. So this gives us a hint, at least, that some of the functions of what UB3A is doing in neurons in the brain might be changing in development, and this is something that might be very important for us to look at. And this information, while very basic science, can form a foundation that can help guide the other therapeutic approaches move forward in a very knowledgeable, rational way. Next slide. Now, back. So I'm going to give you one other example of a basic research finding before I go on into the more clinical work that we do. And that's that we're also trying to determine what happens in the brain when in the absence of UB3A, as in the case of Angelman syndrome. So study in a very detailed way what changes in the brain because of the loss of UB3A. And if we understand this in great detail, it might also help guide therapeutics. Next slide. So I need to give you a quick primer in the most basic sense of how the brain works. And basically, we have cells in the brain that are called neurons that communicate with one another, that they have activity, and they allow us to process information and to think. So this is just an example of 2 neurons talking to one another. Okay? This center of the neuron is called its soma, or cell body. It then transmits information down an axon, which you just think of this as an emitter. So it's like a radio emitter that's transmitting information. It then gets down to a very special juncture called the synapse. There's actually a gap between one neuron and the other. And this is the point at which the signal goes from an electrical signal to a chemical one. So when people talk about neurotransmitters in the brain, it's a way in which a signal can go from one neuron to another neuron. And this information is received on dendrites. So this is a receiver. So one neuron sends information, emits it down this axon. It's an electrical signal. It's transferred to a chemical signal at a synapse, which is a physical gap between 2 neurons. And then the information is received on dendrites of the next neuron, and then the information is passed down along to the next one. Okay? Next slide. Now, I want to basically walk you through what happens when, across this specialized juncture between 2 neurons, how that communication is made. So at the end of one neuron is the axon terminal, okay? And it communicates across this gap to the dendrite on the next neuron. So how does information electrical signal get from here to there? Well, if you advance the slide, packaged in the neuron, which is called the presynaptic terminal because it's communicating then to a postsynaptic terminal, are neurotransmitters. So when you see these fancy ads from the pharma companies, they're always talking about your neurotransmitters in the brain are messed up and you need this to cure your depression. You know, they're talking about neurotransmitters and the signal, the chemical signal that's found in the brain. Now, an electrical signal can come down and invade this terminal. And what happens when that happens, next slide, is it can open receptors on this presynaptic terminal, next slide, that then cause an electrical signal to cause the release of these neurotransmitters into this synaptic space. Next slide. So the neurotransmitter will spill out and then can attach to receptors on the neuron, next neuron. That will open and activate these receptors that allows current to flow in, and then you'll get a signal on this neuron. Now, that's not the end of the story, because you can imagine that if you keep on releasing neurotransmitter, then you will deplete all of your neurotransmitter. So the neurons have to have a way to replenish this neurotransmitter supply. So what happens is these little structures here, which are vesicles that are filled with the neurotransmitter, come back into this terminal, and then they, next slide, they'll eventually get filled back up with neurotransmitter, and you can replenish this supply. Okay? So these vesicles that hold all the neurotransmitter are recycled. All right? And if not, then you won't be able to maintain normal synaptic transmission in the brain. Okay? Now, next slide. We looked at very, very great detail at what might be happening at the synapse in the Angelman syndrome brain. Once again, this is basic research, and this is something that's not immediately going to lead to a therapeutic, but I think you'll see that I think it has the potential to guide very rational therapeutic strategies. So next slide. So this is an extremely high magnification view of a synapse using a technique called electron microscopy. Okay? Sorry, go back one. Now, we've color-coded it, so this kind of pastel-y green is the presynaptic terminal with all these little dots in it, and then it's adjacent to a postsynaptic terminal. Now, when we compared, and this is just a snapshot, we compared thousands and thousands of synapses. When we compared the synapses in a neurotypical mouse brain versus an Angelman syndrome mouse brain, we noticed something extremely striking, and that was an accumulation of what we would call clathrin-coated vesicles. So you can see one there, you can see another one there. At a higher magnification, you can see them here. Now, once again, to a parent, you might not understand what the significance of this is, and I'm going to try it in the next slides to walk you through it. So I showed you how synaptic transmission, how it can proceed normally. Now, if you look in the Angelman syndrome brain, if you advance again, you can get release of neurotransmitter just like before. Okay, next. And next, and you activate neurons. But what happens? Something's going awry in this process where these vesicles are forming, but this so-called clathrin isn't coming off to allow the neurotransmitter to refill in the vesicles. So you get an accumulation of these empty vesicles that don't have neurotransmitter in them. Okay? So next slide. So eventually this will deplete your supply of neurotransmitter back to be released. Okay? So by seeing this accumulation of these empty vesicles, it gives us a hint that the refilling process of reinstating the neurotransmitter at this particular type of synapse in the Angelman syndrome brain, that is one of the things that's going awry in the absence of UB3A. Now, once again, although that was a basic science discovery, now that we have this knowledge, we might be able to target therapeutics that can overcome this deficit, either by finding a way to improve this process to allow these vesicles to be filled back up with neurotransmitter, or to directly reinstate or support the remaining neurotransmitter release that's there. Okay. Next slide. So that just gives you a little bit of insight into why we do some of these basic research studies that we do. And once again, these tend to be supported more by the NIH and less by the parent organizations, but I want you to understand why we do them and why they're important. Is that relatively clear to everyone? Okay, great. Because you're not asking me those questions. Remember, I told you, if you don't understand something, let the questions flow, because a goal is obviously to keep all of you with me so that you're following the presentation. Now, next slide. Now to move back towards the drug development, which is traditionally what the parents and families and caregivers have been most interested in. At least for the therapeutic approach that we focus most of our efforts on, the strategy relies on the basic biology underlying Angelman syndrome. And as you all know, that Angelman syndrome is caused by genetic defects in the UB3A gene. Now, the UB3A gene is actually very unique compared to most genes in the body, with the exception of the sex-related sex chromosome genes, in that usually you inherit 2 gene copies. You can get 1 gene copy from your mom and 1 gene copy from your dad. And those genes kind of share responsibility in producing the proteins that are necessary. Now, the UB3A gene is unique in that it's only expressed from the maternally inherited copy. The copy inherited from the dad, the paternal copy, is silenced. Okay? So it's silenced through a complicated epigenetic process. But the father's copy, although intact, it's silenced through what's called an antisense that's expressed on the paternal side. Okay? So if you get a deletion or mutation in the maternally inherited copy of UB3A, it's sufficient to cause an absence of UB3A gene expression in the brain. And you can see this in the next slide, where this is a mouse model that's used to study Angelman syndrome. And this is what we would call a wild type or neurotypical mouse. And once again, green is the stain for UB3A through a section of the brain. So this is just a section kind of taken ear to ear in the mouse. And you can see that UB3A protein is probably pretty important because it's expressed almost everywhere throughout the brain. Okay, so it's very ubiquitously expressed. Now, you can show very simply that the UB3A protein is coming from the maternal copy because tricks that we can do in mice that obviously aren't feasible in humans but occur naturally as in individuals with Angelman syndrome is we can genetically delete the maternal copy of UB3A. When you do this, you can barely see, but this is a stain for UB3A in the mouse. You don't see the green. Okay? And we've only, go back please, we've only deleted the maternal copy. And what this tells us is that the paternal copy of UB3A is silenced. Okay? So all the UB3A protein that's being expressed is coming from having an intact maternal copy. Okay? So this is really, really important that you understand this basic concept. Is that clear? Yep. Okay. Next slide. So with this biology in mind, we undertook a very ambitious drug discovery program to try to develop Angelman syndrome therapeutics. And I should point out that this approach went well beyond my own expertise. So even though I've trained for more years than I care to admit to be a neuroscientist, I still have a relatively narrow range of abilities, as most neuroscientists do. So I rely heavily on collaborations with people that complement my skill sets, abilities, and knowledge. So for this particular project, I gained, if go back one, sorry. I gained a lot of expertise from Mark Zilka, who has expertise in genetics and molecular biology. And we also got a lot of benefit from Brian Roth, who is an expert in pharmacology and drug discovery. In fact, Brian Roth runs the US federally funded drug program to discover psychoactive drugs. So he runs the National Institutes of Mental Health psychoactive drug screening program. So he has incredible expertise that you normally only find in pharma, and we have it at my university in the form of Brian Roth and his large lab. So next slide. Now, the drug discovery approach, once again, relied and was based on the basic biology underlying Angelman syndrome. So if you can see here, this would be a neurotypical instance where we have UB3A protein is expressed from the eternally inherited copy. The paternal copy of UB3A is silenced. Next slide. In the case of Angelman syndrome, there's a deletion or mutation of the maternal copy of UB3A, and the paternal copy is epigenetically silenced, so there's no UB3A protein expression. Next slide. So an obvious treatment strategy that we and others are very interested in is if there's a way to simply turn on the paternal copy of UB3A, then this has the potential to replace the lost expression of the maternal copy. Okay? And this might sound like science fiction, turning on a gene, but there's every reason to believe that that strategy will work. So one of the reasons that we believe that is that UB3A is expressed off the paternal copy of the gene in other cell types. It's really just neurons in the brain where it's only expressed off the maternal copy. So we know that the genetic information from the father's copy of UB3A is intact. It's there, it's in the genome, it's in the chromosomes, and it's just a matter of finding a way to turn it on. Next slide.